going to have Dr. Barnby going first. He is an assistant professor. He works at the Royal Holloway University. He's a neuroscientist, and he has a lot of interest in doing research with young adults. And there's been a lot of conversation with Dr. Barnby if we can do a collaboration between themselves, himself, his team, and Corpal to see if we can run a study, a research to look into our young adults and how we can best understand the challenges and help them. So, Joe.
So it's a real honor to be invited here to speak to you folks. This is kind of the most fun I think I have when I get to present some of this work, because it means that we get feedback from the people that this actually matters for. So thank you very much, Asper, for the invite. And also, thank you very much, Lynn, for swapping around this session. I'm very much the warm-up act for Lynn Paul that's going to be coming afterwards. So hopefully prep you all for the really interesting talk that's coming next. But as Asper said, I'm an assistant professor at Royal Holloway. I'm a cognitive neuroscientist. So I'm really interested in how the brain makes up the mind in terms of how different parts of the brain kind of enact themselves into the way that we think and behave and interact with others. And particularly some of the work that I'm going to be presenting today is kind of a model of some of the work that I'm hoping to continue with Corpal, that me and Asper have been talking about. And it really centering on how the corpus callosum encodes some of the way in which we make decisions, how we think about ourselves, and how we think about other people as well, and how we interact with other people. And so this particular set of work that I'm going to be presenting to you is just a huge interdisciplinary collaboration between your counterparts in America, the NODCC, and your counterparts in Australia, AusDoc. And this project is kind of a big combination. I'm a very small cog in the machine, really. This particular project is led by Linda Richards and Peter Diane, and now myself as a collaborator, I suppose. And we're a big team comprised of neuroimagers, so people that scan people in those big scanners you might have seen in hospitals and look at how the brain functions and the structure of it. We also work with cellular biologists to understand how different kinds of cell changes might change the way in which the corpus callosum develops. We also have computer scientists working with us to make sure that all the tasks that we give to people are kind of aesthetically pleasing and easy to use, and they're not too weird and complicated that sometimes cognitive tasks can be. We're also full of clinical psychologists, of course, which are the people that work with the folks from the charities that we collaborate with. And also computational scientists, and I come in this last category. So I'm really interested in the sort of maths that underlie the way in which we interact with each other, and how that can kind of be used to explain this kind of way in which we think, behave, and interact. So this collaboration absolutely continues, so this is very much a work in progress. I'd love to hear your feedback or your questions about what all of this might mean, or kind of what your questions are. But I thought I'd give you an update about what kind of work we've been doing, and kind of try and give you some insight into the sort of things that Corpal might be supporting when we're trying to understand this very interesting, kind of misunderstood neurodiversity. So Lynn is going to very much touch a lot more on the etiology and the kind of different presentations of CCD. So I'm only going to touch on this really briefly, but as you may already know, this is something that is usually diagnosed radiologically. So you need to have a radiological report to understand if you have DCC or not. There's not really a behavioral, what we call a phenotype, or a behavioral kind of marker that we can specifically pin the diagnosis to. There's some genetic causes, like everything. Whether we're neurotypical or neurodiverse, there's obviously genetic causes and genetic changes which drive the way in which we think and behave, and CCD is no different. But also, importantly, as you well know, the etiology is very heterogeneous. And what I mean by that is that everyone who has CCD is not the same, right? Everyone is very different. There's a lot of different kind of symptom expressions. There's a lot of different ways in which we think and behave, whether we've got CCD or a neurotypical. And this is kind of something that we're really, really interested in, is understanding, are there any commonalities that occur in folks who are neurodiverse? And is there something actually that once we study CCD a little bit more, we can start understanding a bit more about how the corpus callosum kind of underwrites some of this behavior a bit more commonly? And obviously there's also environmental factors from birth up until adulthood that obviously change the way that we all behave, and CCD is no different. So environmental factors do play a big role in the way in which some of these symptoms may develop over time. So I'm really, really interested in what are the effects of a CCD diagnosis on how we think and behave and interact with each other, and what can we learn about CCD from working with those who are neurodiverse and who have the diagnosis and may present slightly differently to those who are neurotypical. And so usually the way in which we try and design these experiments is we find some folks who are neurotypical, folks from the general population, or we have some folks maybe who are part of the families of those with CCD. And then we also have our lovely CCD participants who we very much collaborate with to design this sort of work. So this experimental design itself was kind of produced in collaboration with OSDOC, who are the Australian counterparts to Corpal. And effectively what we're really interested in is collecting a whole bunch of different kinds of behavioral and kind of cognitive markers affiliated with CCD that would tell us something about not just the sort of things that might occur throughout your lifetime with CCD, but also things that might change moment to moment as a result of having CCD. So we're very interested in looking at things like psychometrics. You might have gone through some of these with a psychologist that you may have worked with, or a neuropsychologist, kind of the neuropsychological tests that you may have been part of. And what we can do is we can apply AI or kind of new changes in AI to understand how those things cluster together maybe differently in neurotypical and neurodiverse individuals. We can then have a look at a whole bunch of things that we still don't really understand about neurotypical folks, but also we know are really important in the way that we kind of think about ourselves. This is called metacognition. I'm going to call it metacognition from here on out. How we think about ourselves, how we think about our own thinking, how confident we are in our own perceptions and behavior. How we also build cognitive maps. So when I'm coming into this room, obviously there's lots of information about how it looks and kind of where all the different objects are. And that requires that I'm building some sort of cognitive map about the space that I'm interacting with. So when I go out of the room, I don't completely forget it. I can come back in and I can probably interact with things just the same as I did when I first entered it and remembered what was going on. And so this is a really important function of what we do day to day, and also inherently an important function in CCD as well. But there may be some differences, and we were really interested in seeing if the corpus callosum kind of contributed to these differences. And also a really important thing that we do, neurotypically or neurodiverse, is we explore environments. As I said, I'm coming into this room and I'm trying to figure out the space of it and what objects are within it. But to do that is require some exploration, whether I'm exploring with my eyes and my vision, or whether exploring with my behavior, trying to test things out and seeing what works and what doesn't. These sort of things are building blocks to the way in which we interact every single day, moment to moment, as with other people as well. And inherently, we wanted to understand, can some of these things maybe help us understand the etiology and the kind of diagnosis of CCD so that we can support people better that may otherwise kind of have difficulties in some of these realms. And something that we were flagged with and we added on in the end, and something that I'm particularly really interested in, is social inference, the way in which we interact with other people. And a lot of the folks from OSDOC in Australia really flagged to us that, these other things are kind of important to us, sure, but actually what's really important to us is trying to understand social cues, trying to fit in, trying to have social interactions that don't feel distressing or difficult or confusing. And so we were really interested as well in kind of digging into this cognitive mechanisms of what it is, the kind of procedures that we use to interact with each other, and how this may be altered in CCD, and therefore how we can support folks better who may struggle with that. So I use a thing called computational modeling to get at this. It basically just means I build a sort of engineered mathematical model of how I think this works, and then we look at how it looks in the real world and we can compare them, and that gives us like a really nice theory to have some sort of marker for. And then, as I said, we have a neuroimager in the team, and so what we're looking at, particularly with metacognition, is how that might be implemented in the brain. And so we can have a look at folks who are neurotypical and neurodiverse and see if there's any kind of differences in the brain that may be supporting this cognitive function. So firstly, I'm going to discuss some of the psychometric findings that I talked about at the start. This is something that I'm only going to touch on briefly, and this is basically just trying to find, is there some sort of phenomenological or some sort of hallmark that we find from these neuropsychological and psychometric tests that might collect together in CCD? Is this something that we can define CCD with, because it's actually quite difficult to so far? And so what we can do is we can take a bunch of measures that, again, you may have gone through in the clinic with a neuropsychologist, but also kind of psychometric traits that we would send online to folks who are part of the community. And we gather this all together and we put it into a big AI machine, basically, and we get a really nice kind of thing at the end of it that tells us if there's any commonalities in folks with CCD versus neurotypical folks. And this is kind of the pipeline of how it looks. I'm not going to go into it in too much detail, but basically we find folks from both communities and we give them all the same tests and we see if anything pops out that is actually really characteristic of CCD versus neurotypical individuals. And I think the thing that we found that was most interesting and something that had actually been talked about in some of the qualitative work or some of the interview work that folks from the Australian community had flagged. So I should mention that there's someone in part of the Ausdot community called Marie Maxfield. She's a person with a diagnosis of CCD. She's also doing her PhD on CCD. So she's very much an advocate for the community over there. And something in her qualitative work that she found is, again, as I mentioned at the start, people in the community really just wanted to understand how to interact a bit more socially. You know, was this something that was really important to them? She identified absolutely there was. And so we wanted to kind of test this in the psychometrics and see, actually, is there something about social interaction that is the clearest marker of folks with CCD, even when you're taking into account all of the other kinds of things that are usually affiliated with CCD. So there may sometimes be incorrect autism diagnosis with folks with CCD, even though some of the features look similar, but it's not the same. The subtle nuance differences. Likewise, are there other kind of features, for example, to do with general cognition or trust that may be characteristic of CCD? But when we put all of these things together into an AI, we find that actually the thing that pops out the most, or the thing that most characteristic with folks with CCD, which matches up with Marie's work actually, is that this kind of idea that you're unable to recognize when someone isn't being very nice, basically. This is called credulity, or this idea of someone trying to trick you and you not necessarily being able to notice that that's going on. And so this is something that's been reported a lot from the community that we worked with over in Australia and the US now, and hopefully continuing with Corpal. And we find that this is something that folks with CCD, especially adults, find very difficult to deal with when they're younger, because they often feel like they get into situations where they're unable to recognize when something's going wrong. And as a consequence, often they have quite distressing experiences resulting from that. And so when they're adults, they're very understandably vigilant about that. I think, we can all understand that, whether we're neurodiverse or neurotypical, something not very nice happens to us, we're probably not likely to do it again. And so something that we recognize in the adults that we worked with is actually that this idea of not being able to recognize when people are being not very nice to you is something that really clearly pops out. And now that we have this kind of empirical data, we have something really kind of solid to move forward with. And actually we try to advocate, or at least I should say Marie, and this sort of data has been used to advocate for policy change in Australia to try and make sure that folks with CCD, are recognised by the Disabilities Board in Australia, so that they can then get some support because of this kind of trait that seems to be popping out.
Sorry, can I just ask, what age were the people that you studied for this? Was it when they were fully adults? Is it longitudinal?
Yeah, so that's a good question. So this is kind of cross-sectional, so we very much used folks who we were able to recruit at the time, but it was over 18. So we had age ranges between 18 and 57 for this community. And again, we only have about 25 folks with CCD in this particular sample. So
It's not big enough to split between, say, 18 to 25.
yeah, not yet. It's a good question because, we want to understand how this changes over lifetime. We did take into account, so in the model, you can control for things like age and gender, and you can control for things like educational attainment, things like that. So there's lots of different things that we would love to stratify and split up and understand how this looks across all ages. But it's just very much at the start of that process. So absolutely important question. And we do see that age plays some role, but even taking that into account, you see this thing popping out basically. But yeah, great question. And also, if anyone has questions during this, please put your hand up and ask if anything's not clear. So something else that we looked at was how we explore, how we sample information in the environment. And this is something that is led by the computer scientist Henry Burgess over in the US. And we're currently working on kind of analyzing some of this data, but I thought I'd give you a bit of a taste of what we found so far. And basically this uses kind of a task that just really simply asks you to just sort of sample some information on this test that we give you. We send it to you online. It's really, really easy to do. Just works in your browser. And basically all you're asked to do is decide all of these grey squares, are they mostly purple or are they mostly blue? Or pink or purple, sorry. My colour vision here is clearly terrible. So are they mostly pink or are they mostly purple? And basically what you've got to do is kind of sample these different grey squares until you're confident enough to make a decision, right? Sounds quite straightforward. And in the first half, it doesn't cost anything to sample. You kind of win points basically for being correct, and we give you some bonus money at the end if you're able to get a high score, or kind of every time you get something correct, we give you a little bonus. But the hitch is that halfway through, it actually becomes quite costly to sample these things. So we actually say, well, the more you sample halfway through, it's actually going to start costing you points, right? So you've got to kind of make a balance between how much do I want to explore and how much am I able to make a decision early on, right? There's a kind of a trade-off you need to make. And so what we found is that, I'm just going to summarize this kind of briefly. Please, these graphs are sort of made for scientific publication, so I apologize that some of them aren't perhaps a little bit more straightforward, but I'm going to summarize them anyway. So we see that when it doesn't cost anything to sample these cards, folks with CCD are absolutely the same as folks with neurotypical individuals, right? There's absolutely no problem. People understand the task. That's not an issue. So when you're exploring an environment, there is no difference between CCD and neurotypical individuals. However, when the rules change very quickly and things become more costly, we find that folks with CCD start to struggle to understand that rule, and they start to explore just as much as they did. Actually, you see their score is no different actually from folks who are neurotypical. So there's no reduction in the actual attainment in this task between neurodiverse and neurotypical individuals, but we see that the adjustment to the rules definitely takes a lot longer. And we see this kind of bearing out in the data. And this is something that's important, of course, because as we know, or as you all may know, is that when there's kind of a time pressure, or when there's lots of complex information being presented very quickly, it can sometimes be a struggle to keep up with that information. I mean, this is something that neurotypical individuals struggle with as well, of course, but it may be slightly accentuated in CCD, and this is something that we're finding. So next, I'm going to be talking about how we think about ourselves, this metacognition I talked about at the start. How do we know when we've made a correct decision or not? Okay, something that is hard at the best of times. If we're tired, we get worse at it. If we've had a drink, we get worse at it. You know, if we haven't had our coffee in the morning, we get worse at it. So this is something that definitely varies throughout the general population as a normal thing, right? So this isn't something that you either have or you don't. It's just something that changes all the time within us, and also may be different from person to person. And this is something as well that Ryan over here in the top right is imaging. So we're looking also at how the brain may encode some of this behavior. So this is very much an exploration into neurotypical and neurodiverse individuals. It's not just a focus on those with CCD. But basically, what we ask you to do is make a judgment call in this task that we give you of whether these dots are moving to the left or to the right. And what we do is we manipulate the dots so they're either noisier or they're more coherent. And that means that we get a really nice kind of graded test of when things become harder in the environment, how well are you able to differentiate that, right? So that's just kind of the first part of the task. It sounds very similar to the previous task I showed you. But the difference is in this one, we ask you, how confident are you in your decision? So are the dots moving left? Maybe you're really certain about that, okay? You're really, really clear. The dots look like they were definitely all together and all moving to the left. How confident were you in that? And so by gathering these confidence judgments, we can actually get a really nice estimate of how much you're using that perceptual information, how much you're using the actual coherence of the dots moving on the screen to inform you about how correct you think you are. So by looking at these kind of different measures, we get an idea about how you think about yourself and how much you're able to kind of differentiate between when you're correct or incorrect. And as I said, this varies among all of us and at different times of the day, so it's not necessarily something that is static in time. And again, much similarly to the previous task, we see that those with CCD versus neurotypical controls perform just as well on the task in terms of differentiating the dots. That's not a problem, okay? So knowing if the dots are going left, knowing if the dots are going right, we see that folks with CCD versus neurotypicals are just the same, okay? However, much like the other task again, when we're asking them to do this slightly more complex task of figuring out if they're correct or incorrect in terms of their confidence, we see it flatlining. And what I mean by that is even though they know or individuals know, and we see this variation as well in neurotypicals, when folks know that the dots are going left or right, they can differentiate that, but then calibrating how confident they are in themselves seems to be a lot different in those with CCD. It seems that it's a lot harder to look into yourself and know if you're correct or incorrect. So that perceptual evidence isn't being used in the same sort of way. And this was looked at in an Australian and an American sample. So we've got some nice cross-cultural data, I suppose, even though, of course, this is all kind of Western samples that we're using. And what we can do is, as I said, we can put this into a mathematical model and we can look at how that looks in terms of what we call sensitivity. And we kind of just have computational neuroscientists like to just have like a sum measure of things because it makes it easier for us. When in reality, things are a lot more complicated. But if we can boil them down to very simple things, at least it gives us some ability to kind of understand what's going on. So it's very much a tool for us more than anything else. But what we see is that neurotypical folks are kind of these blue and red distributions. So you have, as you can see, still loads of variation in neurotypical individuals in how much they're able to notice what's going on and gauge their confidence. But we tend to see that those with CCD tend to be about left shifted. And what I mean by that is that they tend to be slightly less able to calibrate that themselves. So as you can see, they overlap. So it's not that there's a clear difference between groups. These are absolutely different things. It's that they definitely overlap, but it seems that CCDs, or folks with CCD seem to be on the left-hand side of this distribution, so they might be slightly harder to kind of notice when these kind of things are going on. And then lastly, as I said, the thing I'm most interested in, the thing that I'm particularly kind of focused in on in the other kind of work I do in my lab is social inference. And my figure here has clearly become a bit blurry, so apologies for that. That's not your perceptual evidence being wrong. If we're looking at this, I would say that I'm not very confident that I'm seeing this image properly. But I'm really interested in how we build models of other people, right? How do we think about others when I'm thinking about my friends or my family or someone that I've just met here today? I have some model about them in my mind. I have some assumptions. I have some idea about their intentions and their beliefs. And we do this kind of seamlessly, right? We do this all the time. We do it even without thinking sometimes. We just begin to think about maybe, what my friend doing today? I don't know. This is a seamless thing, and yet it's a very computationally complex thing, actually. We do it intuitively, and yet it requires a huge amount of computational energy to actually do this seamlessly. And you kind of notice when we're tired, or maybe when we're stressed out, you notice when we start misinterpreting things, whether you're neurotypical or neurodiverse, you can notice the sort of things that start to go earliest when we're stressed out. It's these kind of social inferences. It's when we start misinterpreting what's going on. And so what we do is we basically get a task where we ask individuals to kind of make some decisions for themselves, and we get a good idea about who the individual is. And then we ask them to learn about someone else. And just by looking at those two things, we, again, putting it into a kind of a mathematical model, we can look at how much you're able to move away from who you are to accommodate someone else, right? And actually, some findings that we see in neuroscience is that actually when we start learning about new people, maybe when you meet new people today, the usual thing is that you assume they're like you, okay? And there's nothing wrong about that. It's just a normal thing that humans do because it's a very cheap way of kind of just immediately having a model of someone else, right? We're all very kind of egocentric in that sense. And again, not in an unhealthy way. It's a very healthy thing to do because it means we're being efficient. But what we can do is we can look at how able individuals are to step away from themselves to learn about someone else, how quickly they can do that, how flexible they can do that. And particularly, and as I said at the start, something that folks with CCD that we've been working with have flagged to us is that they find it really hard to learn about someone else when they're not being very nice. And so we have some people here that you may play with who aren't very nice at sharing points. And what we're really interested in is to see if, A, individuals who are neurodiverse have the same kind of preferences as those who are neurotypical, if they kind of make the same decisions in the same way, but then also if they're sensitive or less sensitive when they're matched with someone who isn't very nice, and if their learning process is a lot slower than those who may be neurotypical. And this is important because if we get this sort of evidence borne out in data, we can take that to individuals who are constructing policy or constructing kind of support options to say, look, this is something that we've really found that's important. We can see it in the numbers, we can see it in the data. This is something that we've tested extensively, and therefore there should be some more support around this. So this is all with the aim of trying to develop more support options for folks who find this more difficult. And this is, I can't remember why I put this slide in, but this is just an example of some of the output that we get from these mathematical models. We get these nice belief distributions that tell us how things change and shift over time. Not particularly informative, I realise, but something that I thought was just good to illustrate maybe what this sort of stuff looks like for us day to day. But importantly, we're still this is very much a work in progress. This is very much work that's ongoing, something that's really important to us, and something that we really want to work together with Corpal as well with, because at the moment we've got OSDOC and NODCC working with us, but of course this is an international collaborative effort. And so what we really care about is working with folks from Corpal, carers, parents, individuals with the diagnosis, so that we can understand what it is that's important to you to look at, and also so that we can kind of understand in your community if these sort of things are also showing as well. Because that might mean that we can collect all the data internationally and have a real kind of global effort to understand this very misunderstood diagnosis. So my email's here. I would love to be in touch with anyone who is interested in being part of these studies. They're ongoing now. The ethics has just been approved. We've been talking with ASPR about how we can start recruiting individuals for these same experiments. And they're very easy. They're online. You just send them. You can do it at home. We don't require you to come in at all to the lab. Of course, unless you want to and you want to come see the scanners and stuff, you're more than welcome to. We're just in Richmond pretty much. But we'd love to hear from you. We'd love to hear your thoughts, hear your qualms, hear your queries, and have you part of this, which is really, really important to us as scientists. So thank you so much. And again, this is not just an isolated effort on my lab at all. This is a huge collaborative effort, and none of this could really happen without all of the different moving parts within this. So thank you very much for listening to me. I'd love to hear your questions.
Thank you, Joe. So I have a question. It's very interesting what you're presenting, in that these individuals may have a bit of difficulty, sort of maybe perhaps understanding social cues, or understanding when someone is being tricky or mean to them. By individuals, I mean, us, our children, the young adults you're dealing with. So I'm wondering if, let's say with repetitive experiences, like life brings them, does that make eventually a difference? Do they eventually learn to pick up on the cues because they've seen it enough? They can now understand the behavior enough, even if it's, every mean person will be slightly different to them, I guess, just like we've all experienced this. But I wonder if eventually they pick up enough information through life, through experiences, and that task may eventually become a little bit easier and they can handle things slightly better.
So this is something that we were talking a lot over the last 2 days about. We kind of had a meeting trying to discuss some of these things that you're exactly touching on here, right? Is this something that is inherently fixed over time, or is it something that can be learnt in different ways over the lifetime? And also, importantly, is this, some of the things that we see in the clinic, is this something that is actually being a social pressure change rather than something to do with the diagnosis itself, right? So there's a few ways in which I could answer that, but I think a lot like, even if we're taking someone, even in kind of neurotypical environments, if you've got someone who learns in a particular way, right? We all know at school, there's some people that just learn in different ways than others, right? And if you have the right environment, you can scaffold that individual to kind of help them learn in the best way possible. And I think this is absolutely no different. So, of course, I've just been presenting to you basically things that we find that are slightly altered cognitively in those who are neurodiverse or have CCD versus neurotypicals. But, we have heaps of stories from folks who are part of these communities that have amazing support around them. And then given that support, they're not distressed, they're fine, they interact in a brilliant way in the community that they're within. So I think it takes knowledge, knowing that this isn't something that you can just press against them and turn someone into another person that you want them to be. It's all about creating an environment that helps individuals thrive with the particular quirks and the particular kind of cognition that they have from birth, right? So it's not something that is fixed. Absolutely, there is a way of, understanding how to recognize For example, when someone's not being very nice, how do you learn that? Well, you can learn that over time. Folks with CCD definitely learn that over time because we see that they become very vigilant later on in life because this is something they've had to deal with when they were younger. But it requires those around them pointing it out and just helping them a little bit more in a much more constructive way to scaffold that learning, you know? So it's not like it's well, that's it, you know, it's done. Like all of us, we have our different quirks and different things that we rely on other people to tell us when, you know, if I'm stressed out and I'm not noticing it, I probably rely on my family to be like, you know, chill out, Joe, you're a bit stressed out at the moment, right? And I might not notice that. So it just takes people around you that are there to support and to provide the environment that can help that learning. Yeah.
No, that's great, thank you. And I think also that goes back a step a little bit when they're still at school, before they become young adults. This is where they can get a little bit better support in shaping the social interactions and understanding all the variabilities they will face. And I think that's probably something that maybe the schools in general could do a little bit better beyond just the academics, because that is what's going to scaffold them and build that knowledge and confidence and understanding, you know, the metacognition, understanding themselves, you know, what do they think about their own thinking, and how does that translate into their young adulthood?
Yeah, and I think this is why, you know, policy advocacy is so important, because without the knowledge that this even exists, how can you know what to look for? How can you know how to support individuals with this? Yeah.
Thank you. Anybody has any other questions? Yes. This is Karima.
I was just intrigued to understand the experiment that you ran on metacognition about the dots, the left and right.
Yeah.
What was your underlying reason for doing that? And I have a reason to ask you that, which I'll come on next, but let's hear you first, please.
So there's a series of, as I said with social inference, there's a series of things that we do all the time, every day, intuitively, without even thinking about it. But actually, the kind of machinery in our brain that is producing those sorts of experiences are quite complex, and they are quite expensive, right? So when we're actually making these judgments about ourselves, we do it all the time because we're probably used to it, but actually it's quite an expensive thing to run in terms of you know, computational power, right? So if we're thinking about something that facilitates that, for example, like the corpus callosum, a really dense white matter tract that allows information transfer, and this is a very expensive cognitive resource that we have, we were trying to ask, basically, if you haven't got that information transfer in the same way, does that mean that these more kind of intuitive but complex tasks start kind of, you notice differences in them, basically. So we're just trying to, you know, it's teaching us about, all of this stuff is teaching us about the brain, neurodiverse and neurotypical as well, right? So we're just trying to understand what parts of the brain are most important for these very intuitive but complex functions that we have. Yeah.
Lovely. So I go into schools and I assess children for dyscalculia. And with one or two children, I observed that they had difficulty with telling the left from the right. So I said, oh, take me to the office now and direct me. And they couldn't tell the left and the right. And I somehow connected this to that practical day-to-day experience that they have about giving directions or getting to a particular place. And that's what intrigued me.
That's really interesting. So that also might be talking to something like laterality, right? So obviously our left and our right hand are kind of wired the opposite way. So my right hand is being controlled by my left hemisphere, my left hand being controlled by my right hemisphere, and that requires communication between the two. So there may be two things there, right? There might be the recognition of which side am I using, and then also not noticing that I've made a mistake there might be changed. So yeah, really interesting observation.
And I felt that, following on Asper's comment, that we can scaffold and we can teach the children how to pick up these skills. But that comes down to the teacher understanding that it needs a very individualized intervention. But there are so many aspects of it that build on it. Thank you. That's amazing.
Thanks for your question. Yeah, it's really important.
Sorry, thanks for your talk. Just wondering, out of everything that you've had and that's been peer-reviewed, has there been any analysis by those that have had kind of early intervention or occupational therapy? I know obviously we've got Nicole's talk later, but I know base sizes are small, it's all kind of at a specific time. But yeah, it'd be great if there's any documents that have kind of looked in that you've seen that have fed into this review.
So, sorry, your question was...
have you been able to split kind of any of the cohorts by those that have had intervention, occupational therapy support, or anything else as dependent variables on the outcomes?
Yeah, I think that's a good question. I don't think we've, at least from our work, you might hear about that later with the other speakers who are also doing fantastic work in this field, obviously. I've come into this field maybe the last 3 or 4 years, so I'm very new to it. I'm very much a newbie. But as far as I'm aware, there isn't any longitudinal work looking at impact outcome relationships in those with CCD in terms of what you're talking about in stratified samples. Something that we really want to look at, but it requires big populations, right? So this is why we're so advocating, please work with us to understand this, because without the involvement of folks from charities like yourselves, it's really hard to study that. So I think tracking that stuff is so vital, and also looking at how those sorts of interventions might improve or change these sort of outcomes, right? That's definitely a avenue for future research for sure. Yeah, thanks for your question.
Any other questions?
Hiya. When you was explaining maybe some of the different causes of the CCDs, like maybe whether it's an isolated case or part of a genetic syndrome, is that, you might have answered it already talking about the level of data, but do you see that play out in different outcomes as part of this study?
So Lynn is going to talk about that a lot more after me in terms of how the different causes of CCD may come about. But in this particularly, we worked with folks who didn't have intellectual disabilities, which usually comes with a particular etiology. We worked with folks who were able to communicate with us and work with us and come in and kind of, some of them didn't live independently, but they definitely were more high functioning, I would say, than some others in the community. So there's definitely a bias in this sample, because we kind of had to trade off practicalities of how can we work with folks who are able to consent and engage and be part of that. We obviously don't want to work with those who didn't consent or didn't understand. So I would say this is probably not, as far as I'm aware, going to be single gene causes for CCD. This is kind of syndromic. We're not quite sure of the reason for the diagnosis, yeah. Thanks though. It's good, again, good questions. These are all really good questions.